Thanks again for joining the Antisense Therapeutics investor webinar. All participants are in a listen-only mode. There will be a presentation followed by opportunity for questions from participants. Please type any questions into the panel on your screen and these will be placed in the queue for the Q&A session. Presenting on behalf of Antisense Therapeutics today, we have the MD and CEO Mark Diamond, Director of Clinical and Regulatory Affairs Nuket Bassem, Director of Drug Discovery Dr. George Takis, and the company's US-based Medical Director Dr. Gil Price. The presentation today will last for approximately 20 minutes, with approximately 10 minutes thereafter for the Q&A session. Bear with me while I bring up the presentation and then I'll hand over to Mark to kick off. Mark, go ahead.
Oh, thanks, Matt. Good morning, everyone, and good evening to those joining us on the call from the Northern Hemisphere. And thank you all for attending today's investor webinar. It has been a very lively and highly rewarding period for the team at Antisense Therapeutics, culminating in two very significant announcements this week. Matt, could you move to slide 3 then in the presentation? Next slide, thank you. Yes. So this week we were very pleased to report that the FDA had granted rare pediatric designation to H1102 for the treatment of DMD. The designation allows us to potentially benefit from what is known as a rare pediatric disease priority review voucher on approval of H1102 for DMD in the US. This voucher presents as an opportunity for an expedited review by the FDA in the process of approving H1102 for DMD. But of course, these PRVs can also be sold, and we know that since the institution of this program, these PRVs have been sold for between 68 million and as high as 350 million. So we think that the rare pediatric disease designation is a great endorsement for our program, recognise the need for improved therapies for rare diseases for children. And we were very, very excited to be able to advise our shareholders of this potential opportunity to benefit from the work that we're doing in Duchenne's. Next slide, thanks, Matt. But today we're looking to report on the next piece of very significant news for Antisense Therapeutics. We alluded to this earlier in the week that we have new data presented just last night at the World Muscle Society. It's one of the biggest congresses held in the world of neuromuscular disease. And we were very, very pleased that our poster was presented at the conference to people in the DMD community, and the data that was presented at the conference. We'll go through in more detail in the presentation today. Dr. George Takis and my team, he will be taking through the findings. The data, oh, sorry, Matt, thank you. The data that we presented showed that our data that we reported on our H1102 phase 2 clinical trial, when we compared it to a matched natural history control population of boys, we were able to show our drug with a statistically significant improvement in total pool 2 scores. That's an assessment of muscle function compared to this natural history control. So we think this new data both elevates the quality and the clinical significance of our data. We think this is data that's going to be enormously helpful for us in the continued development of this drug. We think that the fact that this data has been presented at one of the most significant scientific congresses in the world of DMD is further sort of validation for the import of this data. And so I'll hand over to George, though, to take you then through the specifics of the data that we presented. He'll conclude on that data, and then we'll be keen to take any questions that come through from the audience. Thanks, Matt.
Thank you, Mark, and welcome everyone. This is George. Antisense Therapeutics pioneered the use of AT1102 to treat the inflammatory muscle damage in DMD. And if we move to the next slide, as this slide indicates, AT1102 is an antisense drug, and it's the CD49D. And a scientific publication, the one on the right, indicated that CD49D is a progression and severity marker in DMD. And the Antisense team conducted a successful phase 2 open label trial with 1102 in 9 non-adolescent ambulant boys with the Royal Children's Hospital in Melbourne here. So the boys in our trial took AT1102, as you're all aware, for 24 weeks, 8 on top of their corticosteroid dose, and 1102 appeared to be safe and effective in modulating the target, CD49D, and also demonstrating positive effects on the multiple disease progression parameters, which we've previously talked about. The boys maintained the performance of upper limb function and the strength and the muscle structure by magnetic resonance imaging, and that's compared to the literature reported losses for the boys on corticosteroids. Next slide, please. Our objective was to better assess the quality of the effect of the performance of upper limb function, PUL2, because that's the registration efficacy endpoint for drugs in non-ambulant boys. And we did that by comparing AT1102 PUL2 data in the phase 2 trial with a matched natural history control, an external control. Next slide, please. This figure shows the total PUL2 measure and the 3 dimensions of the upper limb function. From the top, you see the shoulder, and there's 6 items. The elbow, where there is 9 items in the mid-level, and at the bottom, the wrist and hand with 7 items. And then there's a separate item, which is called the PUL2 entry score, which is not part of the total PUL2 score, but it's also measured at the start. The maximum total PUL score one can achieve is 44. Next slide, please. So we teamed up with Professor Eugenia Mercuri and America Payne and Giorgio Carradi in Rome, Italy. And I was on the call, as Mark indicated yesterday, actually this morning, with the poster presentation. And they're experts in PUL2 and treating boys with DMD. So they have a natural history database of boys with DMD, and they use the inclusion criteria we use to enroll the non-ambulant boys in the 1102 phase 2 trial, and they found 20 matched boys from their natural history database with those inclusion criteria. Next slide, please. So this is the data. The baseline data indicates the demographics of the age, the steroid use, and the PUL2 results of AT1102 on the right versus the Rome cohort on the left. And they're similarly matched for age, frequency of steroid use, and the total PUL2 in both groups. So as I indicated earlier, there's 20 patients identified by the in Rome cohort, and there was 39 assessments at 24 weeks. And we had our own phase 2 data with 9 patients and 9 assessments at 24 weeks. So of those assessments in Rome, 37 were on steroids and 2 were not on steroids. And in our case, 8 assessments were on steroids and 1 not on steroids. So that's a similar frequency. If anything, ours had fewer patients on steroids, which makes it harder to treat. Age at baseline was very similar, 15.6 in the Rome cohort versus 14.9 in the AT1102 cohort. And the total pool 2 mean score at baseline was 20.2 for the Rome cohort and 24.8 in our cohort. None of these were statistically significantly different, but that measures the baseline scores. And I'm pleased to say that I've presented the work as this morning on behalf of the Antisense and the Italian teams at the World Muscle Society. And if we can go to the next slide. And as the poster read, the good news was that AT1102 improves pool 2 in the non-ambulant boys with MB compared to the natural history external control. So I'll now take you through the results and the important results, comparing the 24-week AT1102 phase 2 pool data to the match run cohort. Most notably, you'll see that the AT1102 treated patients demonstrated a statistically significant improvement in the mean pool 2 scores for the 24-week treatment compared to the matched Rome cohort. The AT1102 achieved a total pool 2 score of plus 0.89 over the 24-week. That's the mean plus 0.89 compared to a mean of minus 2.00 in the match Rome cohort, with a p-value of 0.01. So highly significant there, and the differences is quite high. The match, the forest plot just below, it shows the 1102 24-week treatment had a mean increase from baseline in pool 2. You'll see everything's above the 0 line. It's indicating the standard error of the mean minus the the mean minus the standard error of the mean, and the mean plus the standard error of the mean. And you can indicate that how far data would vary from the natural course. That's what the standard error of the mean does. So the Rome cohort 24-week data shows a decline from baseline in the pool 2 mean minus 2, with the standard error of the mean always being negative, which is consistent with the published one report I've seen published on pool 2 at 6 months, and other reports on pool 2 published that I'm aware of. So this is highly exciting data and consistent results with the control, where you just don't see that all negative, and you do not see those improvements with other drugs in development. Next slide, please. Slide 9, this is slide 11, sorry, shows the percentage of patient responders in AT1102 study compared to the Rome cohort. The 1102 treated patients had a higher percentage of patients with an improvement compared to the matched Rome cohort in the pool 2 parameters. That's all of them, in fact. So, and it was, for the total pool 2, it was 44% improving with AT1102. That's actually improvement versus 18% improving with the external control. And the frequency of patients showing either an improvement or no change, which in itself is beneficial to patients compared to what's happening at the moment. The score was 78% improving or stabilizing with AT1102 compared to 33% only in the external control on standard of care. So a lot of boys are going to benefit in the upper limb function using AT1102. Next slide, please. So, thank you. In conclusion, 1102 achieves statistically significant gains in the total pool 2 score at 24 weeks compared to the standard of care, mostly corticosteroids, in a matched cohort of non-ambulant DMD patients, and with a 78% frequency improving or maintaining their total pool 2 score. And that significant gain in the mean, to my knowledge, I have not seen reported anywhere with any other drugs, though I have seen the similar losses that we reported today. There's importantly, there's general acceptance for the use of total pool 2 as a primary efficacy endpoint for registration studies in non-ambulant boys, and that's because it's a robust marker. So we are very excited and looking forward to utilising this new comparative pull to external control data, and our previous data showing those positive effects on the strength and MRI muscle structure in advancing development of 1102. And then with all this data, we sort of we think it's driving hope that 1102 will make a significant difference in boys with DMD. Thank you, and happy to field any questions.
Thanks, George. Mark, were you happy to carry on to the Q&A?
Yeah, thanks, Matt.
No problem. Thanks again. And if anyone would like to submit a question, please type into the panel on your screen. The first question is goes as follows. Natural history data suggests that the rate of PUL 2.0 decline is steeper for boys who enter studies with a higher baseline measurement. When you look at the best and worst responses in your phase 2A study, was there any correlation with baseline, or was stabilisation/improvement independent of entry measurement?
Thanks, Matt. I'll get George to address that if you could, please.
Thanks, Mark, and thanks for the question. Yes, we, using the pull to entry score, which is the way patients are measured as to their level of upper limb function when they enter the study, there is more often than not loss is greater when there's a higher entry level pull to.
Thanks, Matt. Next question.
Sorry about that. Just trying to get off mute. Did the PUL 2.0 responses in your study correlate with changes in lymphocyte populations expressing CD49D? If you could please relate today's information back to the drug's mechanism of action, that would be helpful.
I'm happy again to answer that question. Thank you. So that is the question that we set out to answer as well. And as previously we've presented, we've shown the individual patient pull 2 data, where 7 out of the 9 patients had stabilised or improved. And we identified that those patients during treatment versus post-treatment, when we saw post-treatment escalation of CD4 positive T cells post-treatment, those are the patients that had stabilised or improved. So that is suggestive that the drug is modulating its target and working by a CD49D T cell response with regard to upper limb function.
Great, thank you. The next question is, could you please comment on the relative stabilisation and improvement you saw between the different components of PUL 2.0, shoulder, elbow, distal, to help us understand the clinical impact? Do patients tend to lose those functions sequentially or at the same time?
George, you okay to grab that?
I was looking at that data at the poster, I must admit. Yeah, so we identified an increase in the mean of the total pull 2, as I've indicated before, but also that was 0.89. We had a similar increase in the shoulder component, the mid-level elbow component, and also the distal hand wrist component. We did have many increases along each of those parameters, as well as the pull to entry score. And that's individually, patients tend to lose their ambulation first, and then they become non-ambulant, and gradually they lose the shoulder function, followed by the mid-level function, and the hand wrist function in time, and then in longer time then it takes to lose, sadly, respiratory problems and then cardiac problems. That's probably the order of things. And so by us and our compound maintaining the the total pool 2, and also maintaining the different components, it's very important to these boys as they're at different stages of their condition.
Great, thanks, George. The next question is, are there any restrictions on who you can sell the RPV voucher to?
I'll address that one. Thanks, Matt. I don't believe so. I think that any company that wants to, or has interest in purchasing the voucher, is a candidate. So no restrictions, as I understand.
They're ringing you up now, Mark.
The next question is, are the 9 boys continuing with ATL 1102 treatments?
Maybe I'll get Niket to address that, if you could, Niket.
Thank you, Mark. Good morning to all, or good evening to others. Once the study was concluded, we did not continue treatment of ATL 1102 in these boys. It was primarily because of the approval that we had obtained initially for the clinical study with the ethics committee. We contemplated continuation, but because there was such a large gap between the first and last patients, it became a little bit tricky at the end to restart the study. It would need to have come on as a new study, obviously. But we are obviously, with the progress into the phase 2B study, we are obviously looking at including as many boys as we can to give them opportunity to have access to the drug.
Thanks, Niket. The next question is, Mark, can you tell us about the status of the US legislation to extend the RPD voucher program beyond its current expiry in 2022?
Yeah, thanks, Matt. Yes, there is legislation that's I think now been approved by the House in the US to extend the RPD voucher by a further 4 years. So my understanding is now it is to go to the Senate to be cleared. I understand that the biggest roadblock was getting the House to approve this new legislation. And so now just waiting for Senate clearance. That would add 4 years to the program, meaning that we would have potentially to 2026 to see ATL 1102 approved for DMD and benefit from that, potentially benefit from the PVR.
Great, thanks, Mark. Next question is, while I can see the results are clearly solid, I struggle to imagine witnessing the in-person changes you describe. Can you provide a qualitative descriptive view of the results? What could they do on drug that they couldn't do off drug?
So maybe I'll address that question initially, and perhaps just give Gil Price a moment to also sort of contemplate it, and George and Niket. I can't speak specifically for this data, and the others may do, but what we know is that once the boys lose the upper limb function, it's not able to be regained. And in interviews that we've conducted with the non-ambulant boys, we know that the loss of pool 2 function that we're describing here can be the difference from boys being able to brush their teeth, to being able to be using iPad, and to be able to feed themselves. And so, a change of the scores that we've seen are what the physicians are regarding as clinically significant, because they bring those type of changes for the patients. And when the pool 2 assessment test has really been designed to be able to to be able to monitor those types of practical changes for the boys and functional losses. And so, that's why the data is being described as clinically significant, because it is having, that type of benefit on the boys to allow them to be able to see that those type of quality of life improvements. But Gil, would you be comfortable just giving a perspective on that as well?
Sure. Yeah, so thanks, Mark. So not only would I concur with that assessment, but I would maybe add to it and maybe make it a wee bit more dramatic in this way. I have been very close to the advocacy groups in Duchenne for a long while now. And so when you talk to them, when you talk to mothers and you talk to some of the boys about these things like being able to use an iPad, brush their teeth, hold a pencil, to fill in a blank on a school homework assignment, etc. A lot of times what you hear is the word profound, that their ability to do this and to do these simple things maybe longer than they might ordinarily have not been able to do under the influence of our drug. They use the word profound. So again, as we think about all of this, I would just encourage you to remember that small things that we may look at in our lives as being small, are profound in the life of a boy with Duchenne. So that's it. Yeah, I agree with how Mark has kind of highlighted it here. The ability to use an iPad, the ability to brush your teeth, the ability to hold a pencil, to fill in a school homework assignment. Those things are significant, if not profound, in this way.
Might take the next question, Matt.
No problem. That is, can you sell the RPV voucher onto any drug company?
Yeah, I think we might have in fact covered that one earlier. Thanks, Matt. Yeah, just conscious of time too, Matt. Maybe kind of last couple of questions, and
sure,
we can follow any up subsequent to the meeting.
Yeah, absolutely. So a couple more. Why were there 39 assessments in the 20 Rome boys?
George, could you comment on that, please?
Yes, the cohort in Rome has been followed for a couple of years now. They've around Italy, they've got 90 patients, and in the Rome cohort, the ones that matched our inclusion criteria there were 20 boys, but because they followed them for more than a year, regularly measuring their Pool 2 scores, they've got about 2 readings from each boy over a 6-month period. And because Pool 2 losses are linear over time within the non-ambulant boys, they're able to provide 39 readings from their database.
Great, thank you. The next one reads, while the differences in baseline PUL score are not statistically significant, do patients with a PUL of 20 versus 24 have any differences in quality of life? Additionally, while the changes from baseline are statistically significant, does that actually mean anything to the lives of patients? i.e., does this improvement actually translate to better quality of life for the patients?
Yeah, thanks, Matt. Look, we have gone some way to address that in the previous query, but maybe might get either Niket or George just to talk about that numerical change in Pool 2, what is regarded as being clinically significant. You happy to tackle that, George?
I'll tackle the question initially on the difference of the means at the beginning. So and I'll perhaps get you to indicate what's clinically meaningful, but we also have a quote to that effect as well in the announcement. So I'll indicate that the baseline demographics indicated a couple of points difference because they just happened to use the boys that they had in the database. But that wasn't statistically significantly different. That was just that was within the standard errors of the mean, and there wouldn't have been any statistical significant differences between the groups. Niket, I'll leave you to, in terms of the clinical levels and the levels of improvement, we've got a score of plus 0.89 over a score of -2. That's a nearly plus 2.84, and that's a huge difference. Normally boys would be losing that much function, but to be maintaining it and then gaining some is, I think Professor Mercuri is quoted, is clinically meaningful, and you may have also more information from other experts like Professor Voigt.
Thanks, George. So when we were having a look at the data and I think I presented some of this in our presentation of the data initially, when we spoke to the experts at the time and looked at the type of changes that we're observing in the boys over that 6-month period, our expectation was, and also looking obviously at this Rome cohort data, which is more closely matched to our patient population, a change of 2 in the pool score, in their overall pool score from baseline to the end at 6 months was considered to be clinically meaningful. Normally these are looked on an annual basis, in which case the expectation is a change of 4 points. I mean, this we need to look into a lot further, but in those initial discussions, that was the feedback I received from the various KOLs. And that was the reason why we sort of looked at our data and assessed where we felt we had clinically significant improvements in the patients. And that was presented and should still be available on our website.
I thought maybe appropriate also, Niket and George, to say that the endpoint of total pool to change over the duration of treatment, is what we understand would be the registration endpoint, efficacy endpoint for a pivotal study. So the data that we're highlighting here, where we're showing statistically significant differences, is the type of data that the regulatory authorities would be looking for to approve a drug for non-ambulant boys. So, today it's what really gives the most, I guess, significance to what we're reporting on today is that we are talking about exactly the type of data that the regulatory authorities would expect to approve this drug. So that might be, just given the time, Matt, I know we've gone past what we thought that we would allow for the Q&A, and I'm sorry for those who've put questions in. I know we've got quite a few that we haven't been able to address now, but happy to follow those up subsequently. But I think just thanking my team, for the wonderful job that they've done in the development of this program to date, and I think culminating in this new data that we've highlighted here, to have been able to bring on interest from Eugenia Mercuri and the team there who to be involved in this program. They are experts in the field and are very, very busy clinicians, but just their preparedness to assist us, I think, gives further validation of the work that we're doing here. So, I just want to take the opportunity to thank George, Gil, and Nuket for their efforts to date. And we're very excited very excited about the continued development of this drug, and just echoing what George said, that we think we've got a program that could really bring important benefits to a population, the non-ambulant boys with Duchenne's, who really have got very, very few options. So thanks, Matt, and might conclude there.
No problem. Thanks, Mark. And for those who put in questions that we didn't get to, we've got your details and email addresses. So I expect Mark and the team will endeavour to get in contact with you by email after this session. But thanks to everyone for joining, and we'll close the session there. Thanks.
Thanks, Matt.
Have a good day.
Thank you.
Bye.